This video is a summary of Part 3, Biochemical Pathway of Complex Formation. In this segment, we delve into the biochemical pathways of complex formation, specifically focusing on the interaction between RNA and various proteins. As we analyze the formation of the VP35 RNA complex, we observe a fascinating trend. As the dissociation constant decreases, there's a corresponding increase in positive entropy change, TS. This indicates a more stable and spontaneous assembly process. The analysis of biochemical pathways for the formation of the VP35 RNA complex demonstrates the co-directionality of changes in calculated physical parameters such as Kd and Ts. The transition from simpler molecular formations to the final product is characterized by a consistent decrease in Kd values and an increase in positive Ts, reflecting the stabilization and spontaneity of the assembly process. Now, let's look at the directions of change of thermodynamic quantities for proteins that bind poorly to the RNA molecule. If the change in entropy as a whole indicates the final product, then the minimum value of the dissociation constant no longer indicates the final product, as indicated in the diagrams. Overall, the study highlights the interplay between entropy and dissociation constants overcoming the assembly of VP35 complexes. The results provide valuable insights into the thermodynamic landscape of molecular interactions and the impact of mutation on the formation of stable biological complexes, offering a foundation for further exploration of protein-RNA interactions.